to imagine this scenario. If someone across from the room from you sprays perfume, after a minute or two, the scent travels to you. Using your best prior knowledge, I want you to explain how that happens. If somebody sprays perfume in one part of the room, and then a minute or two later it reaches you, explain how that happens. Using your best prior knowledge. If you're not sure, just give it your best shot. Share your answers with that person, and I'm going to be listening in as you guys talk it out.
Oh, somebody talks.
Okay. So you're thinking of going around people talking maybe. What's moving? What should that look like? Like sound waves move. The particles spread around. Okay. Somebody want to tell me what you think's happening? If somebody sprays perfume in one part of the room, and a couple of minutes later it reaches you, what's going on? How do we think this occurs? Jasmine, what do you think?
I think it would move like sound waves. I don't know, call it smell waves or something.
Smell waves? Okay. So what do you think is moving?
The waves of like a scent or something.
The waves of the scent? Okay. Molecules? What molecules, Jackson? What do we know? Air. What state of matter is air? It's a gas. Okay, so what do you guys remember last year about how molecules are arranged in a gas in that state of matter? What do we know about them? How do they move?
They're spread out and they move faster.
They're spread out and they move faster. Okay, so how does this occur? How do those molecules from one part of the room get to you? What do you think's happening?
Well,
Okay, so if they're always in motion, eventually they're going to reach you. And someone asked about, or mentioned in another conversation I heard over here, someone mentioned an air vent. Well, would this still occur even if there was an event in the room?
Yes.
Even if there wasn't a draft, would those particles move and continue to move until they eventually reached you? Okay, so as we're exploring this idea today, we kind of have an idea, but we're not real confident about what's going on. So by the end of the lesson, I'm going to have you come back to this question, and hopefully we'll revisit it with a better understanding of scientifically what occurs that makes this phenomenon true. But specifically in our lesson over the next couple of days, we are going to learn how the cell gets materials that it needs to function and to allow a plant or animal to survive. And within that lesson, starting today, we want to be able to define a couple of scientific vocabulary words that connect to how the cell gets what it needs. Before you leave, I'm going to hold you accountable for your learning. We're going to come back to that first question and see if we can answer it. Tell me what you're going to be doing inside of the lab with this smelly balloon lesson. What does it ask you to do with the balloons? You're going to smell it. Okay, at this lab, and when you get to your station, you're going to have 3 different colored balloons that you're going to be observing. And you're going to be observing using your sense of smell. Is that a qualitative or quantitative observation?
Qualitative.
Very good. So we're not going to have a pink balloon at your station. You will observe that pink balloon by smelling it and then passing it to the next person at your station. And as you smell, I want you to try to make a hypothesis, a prediction, about what you think that fragrance is that the balloon is giving off. Now, you guys know the procedure for dismissing into the lab. So as I called your lab group, you're going to go to that lab station. You're going to sit and wait until everybody is seated, and then you can begin your observations. Okay? Station 1, if you'll go to lab station 1, please. Group 2, if you'll go to station 2. 3, if you'll go to... Wait.
It smells like sweet tea. Vanilla lavender, coconut oil.
You guys say, how in the world did that happen? What's going on? Trinity?
The balloon fell and the balloon absorbed the liquid, so.
Okay, well, first of all, is the balloon living? So is it made of cells? Okay, so how is that fragrance able to get from inside the balloon out to you? What do you think?
I'm gonna good guess and say the cell particles are like condensing and putting the fragrance on the outside of the balloon.
They're condensing.
Yeah, like in like a styrofoam cup. Whenever like the water droplets go on the outside.
Oh, okay. So you were thinking they were condensing on the outside of the balloon, but they're inside.
Yeah, but they're inside.
So how are they getting out to you? This barrier, kind of like the cell's membrane, does it act kind of like the gatekeeper? Okay, so I'm still wondering, my thoughts are still, how in the world that happens? You guys came up with some good ideas that those molecules are definitely able to pass across this barrier. But how? How is that able to occur? Okay, Blaze, you want to take a guess?
Whenever the balloon stretches, it makes little microscopic holes and allow them to smell the top of fluid.
Okay, so are there microscopic holes in this balloon that we can't see?
Yes.
There's tiny little pores, there's tiny little holes that allow that gas or that material to leave the balloon. I want you guys to draw your balloon on your paper. Let's draw a little sketch here of what's happening. Here's our balloon. Okay. And here. Besides this experiment, is there any other real life situation that proves this point, that a balloon has pores? How do we know that that's true from our real life? Amy?
When you get a balloon, and then like after a while, like the helium and it deflates.
The helium in a balloon deflates. Has it ever happened with a plastic balloon before? If I left this in the classroom over a week and I came back and I observed it, what would I observe? It gets smaller over time. So we know that this is true from our own real life. You just may not have known what was going on. Has anybody ever popped popcorn in the kitchen and you follow the trail? Wait a minute, hold on. I smell popcorn. Let me find it. Bingo, there it is. So is that a real life example of the same phenomenon? Okay, so maybe popping popcorn. What are some things that a plant or an animal cell would maybe need to take in or release out so that it can maintain homeostasis? What are some things you know that it might need? Water, for sure. Excellent. What else? Tyson? Food. Okay. And in science, we've learned a special word for that food. What gives the cell energy? There was a special word, started with a G, that gives the cell energy. Jasmine?
Glucose.
Glucose. So the cell would need glucose or sugar. Okay. Adrian, you got one?
Just like air.
Okay. So like for an animal, what type of gas would it need to survive? Oxygen. And for a plant, what gas do we think? Carbon dioxide. Excellent. Excellent. And that's kind of where we're going with this whole unit. So I wanted you guys to start. What about anything else it would need to take out? What else would it need to take out so that it's healthy? Waste. Okay. So for an animal, what waste might need to leave the cell? Carbon dioxide. And for a plant, what waste might need to leave the plant cell? Oxygen. Very good. But I want you to continually, throughout this lesson, over the next couple of days, make the connection back to the cell. Don't forget that we're thinking about this process in terms of how the cell gets what it needs in order to survive. All right, guys, as we come into class today, I want you to find your bell ringer question or your warm-up question. And the question says this. John wrote the following journal entry in his science notebook. Explain if John's notes are correct or incorrect. Change any mistakes in his notes so that they are written correctly. So you really have two options here. You can either have an option where you say John's notes are correct scientifically, or you could maybe make some changes to his notes. So I'm going to give you a minute to read that statement that John wrote. Decide if you think that his statement is correct or incorrect. And if there are any mistakes, in his writing. It is your job as a good science student to give him feedback and change his answer so that it is scientifically correct. If you make any changes, be sure to explain why you're making those changes on your paper, okay? Diffusion, or we have equilibrium. Which of those is best describing what's occurring with the perfume in the classroom? Is it diffusion or is it equilibrium? How many of us would think diffusion? Okay. How many of us would think equilibrium? Okay. How many of us think we could include both of those words in our response? Okay, so a couple of us think both. So if that's the case, Lauren, how could we include both of those terms in our response?
Well, equilibrium is when it equals out through the whole room, and that's when it did. And diffusion is when it, like, you spray something at first and it's really strong, but then it, like, goes, like, kind of similar to equilibrium.
Okay, so remember that we saw a demonstration, or we discussed a demonstration earlier this week with air freshener or possibly with perfume, like what's being described here. So if it's sprayed in the classroom where the teacher is standing, where are the molecules in the highest concentration? Where she sprays. So when she sprays it, that's where the molecules are going to be in the highest concentration. And then over time, tell me what happens to those molecules. What occurs, Blaze?
They spread out.
They spread out. What's the science term for that? They diffuse. So they go from an area of high concentration over here to an area of low concentration. And as that occurs, what happens with the fragrance, or what happens with the scent? Trinity, what happens?
It spreads throughout the classroom.
Okay, until what is reached? Equilibrium. Okay, so we would say this is not an example of osmosis. Why is this not an example of osmosis? Josh?
Osmosis is diffusion in water.
That's diffusion of water. Do we see diffusion of water occurring in this example? No. So we would definitely change osmosis to the word diffusion, and then we could even improve it even further by saying diffusion will continue until a balance of molecules is reached, and that is called what? Equilibrium. Until all of those molecules are completely diffused into the room, and they're equally diffused into the room as well. To it. So today I want to just kind of review where we've been this week and kind of talk about where we're going for today. Remember, as we started this unit of learning, on Monday I took you into the lab and we did our smelly balloon lesson. Who remembers what happened with our smelly balloon lesson? What did we kind of learn from that experience? Jasmine, what did we learn?
That the balloon has pores that allows the air to diffuse out of.
Okay, so I had placed different fragrances inside of the balloon, and we learned that the balloon has different pores, or it has a certain type of membrane, like a cell membrane does, and you guys were actually able to smell the fragrances that I put into those balloons and make some observations. From there, we came back into the classroom and we defined what those terms meant. We defined selectively permeable membrane and what that means. We defined osmosis was the diffusion of water, and that diffusion simply anything that's going across that cell membrane that the cell needs. And then we just reviewed equilibrium. Class, what's equilibrium? When those molecules form a equal amount or balance, okay? And we discussed on Tuesday sort of what this would look like if we were to illustrate it or demonstrate it, because I told you our end goal is to be able to do that. We want to be able to draw this process taking place. And so we talked a little bit about what equilibrium would look like. Can I have a student tell me what equilibrium would look like if I were to draw it? What would it look like if I were to draw a cell in equilibrium? Okay, so what would that look like, Josh?
It would be the same amount of molecules inside the cell that are outside the cell.
Okay, what do we want to put on the outside, Josh? 4. All right, so if I had 4 molecules on the outside, in order for that cell to reach equilibrium, how many molecules would need to be on the inside? 4. Okay, and then at that point, the cell has reached a balance of materials. What occurs with osmosis and diffusion at this point? Do we have a need for the cell to move any more materials in or to release any more materials out? So once equilibrium is reached, what happens to osmosis and diffusion? They stop. They stop. They no longer need to move in or out. And this is the way that the cell maintains homeostasis, a balance of materials. Because once it gets that balance reached, it doesn't need anything else. This is how your cells reach in a balance of oxygen and carbon dioxide and all of the molecules that it needs to survive. And then from there, we moved into some research. So I just want to see real quick, Does anybody remember any facts from our research yesterday that we read about some of these materials in our lab? Josh, what did we learn from our research?
When cornstarch is mixed with iodine, the iodine turns black.
Okay, so when cornstarch and iodine meet, the iodine turns black. Okay, Peyton, what else do you remember?
Well, the cornstarch turns black because the iodine has a special property.
It has a special property. Do you remember what iodine was called? Because it turns black, what is it called? Dylan, do you remember? No? Josh?
Monkey blood.
Okay, that was a slang term that we learned, was monkey blood. Well, what do we call something that changes color in the presence of something? It's an indicator. Very good, Trinity. So we learned that iodine's an indicator, and it can indicate for cornstarch. Are there any other facts that you remember from our research?
The cornstarch molecules are too big to fit through the semi-permeable bag.
Okay, so we learned that cornstarch is a relatively large molecule, and it's not easily diffused. It can't get through that bag. Anything else? Lauren, remember?
Iodine has small molecules, so it can go through permeable bag.
Good. So iodine's a small molecule, and class, what does that mean about iodine?
It can go through the bag.
Can get through. So the size of the molecule depends on whether it can enter a cell membrane or whether it can exit. And so from there we developed a testable hypothesis. And our testable question that we came up with was, how will iodine affect the color of cornstarch in a semi-permeable bag? Who remembers what we as the scientists are going to be adding? What are we going to be changing in this experiment? What is it that we're in charge of? What are we adding to it?
Iodine.
The iodine. So what variable is that?
Iodine.
That's our IB. Okay, so remember that was our independent variable, was iodine. And what's the color of cornstarch? That's our dependent variable, because that's what we're going to hopefully be seeing as a color change, right? All right, guys, and I want you to take a look at the top of your lab handout so we understand the overall objective and the overall goal of where we've been trying to get to this entire week. By the end of this lesson and by the end of this class period, I want you to be able to illustrate and demonstrate osmosis or diffusion, okay? And the purpose of this lesson is that we want to be able to hopefully observe what a semi-permeable membrane does as it works to bring materials in or out of it. And so at our stations today, can somebody tell me what our semi-permeable bag or semi-permeable membrane might be represented by? What is it that's going to be our semi-permeable material? Peyton?
The bag.
The bag itself. Okay, so everybody go ahead and find the cornstarch bag at your station. And you can observe that at your station. Remember yesterday we learned about the properties of cornstarch and what it's used for. I want you guys to go to the bottom of your paper and locate your data table. And now that you've looked at your cornstarch, should we be able to recolor? Because you've just observed it. So what would we write as our starting color for our cornstarch bag? What color is your beaker? Go ahead and record the starting color of your solution. So I've gone ahead and drawn on your paper. You already have this. If you'll flip to page 2, I've drawn kind of an initial setup for our lab. And we're going to be using our color pencils and our markers today as we color code each of the molecules that may possibly move in this experiment. So we have blue, large blue circle, that's going to be represented on your model for water. You're going to be using a small red square to represent your iodine molecule, and you'll be representing cornstarch with a green triangle. So that as we illustrate or as we draw what has occurred in this process, you guys can see the difference in those molecules in terms of their size and their color. So our lab setup right now is that we have iodine in the beaker, and we have our cornstarch bag outside. It's kind of like a
There's like some black.
It does?
It turns black, like really black.
Look, there's certain black.
Yeah.
You see the purple?
The water in the beaker. Has there been any change of the color of water?
Yes.
Is it changed from dark brown to any other color? Okay. So what does that tell us about the cornstarch molecule? Okay. Did we have any observable changes in the color of the water? So iodine's still in there, water's still in there. Did we have any movement of cornstarch? No evidence of that. So kind of like, I'll say purplish or a blackish color. Some of you had dark black, some of you had a kind of a purplish color. What would you infer would the bag look like if I left it in there overnight? It would be totally black, right? Because it takes time for that process to occur, okay? What would happen if I placed my beaker on a hot plate during this experiment? Maybe I gave like group 7 a hot plate and I let the others of you have your beaker on your station. What would occur if I placed that on a hot plate? Josh, what would you think would happen?
The reaction would happen quicker.
Why would it happen faster, Josh?
Because the molecules are heating up, which makes it move faster.
Very good. So when we heat an object, we know that those molecules move at a faster rate. So does that mean that the diffusion would happen at a faster rate as well? Okay. What about if I put cold water in your beaker instead of room temperature water?
It'd be slow.
Okay, so we would infer, so we could even design a whole new testable question just from that one change, couldn't we? We could design a whole new experiment just from that one change, just the temperature of the water, okay? Can we think of any other ways that we might could change this experiment? If we wanted to test it again, is there any other way that we could change this experiment and maybe come up with a new testable question? Anything else you could do differently? Matthew? Different bag, okay? A thicker bag, a different texture, or maybe a different type of bag, Lauren?
Change the density of the iodine.
The density of the iodine. What do you mean by density of the iodine?
Like put more, like take out less water and put more
Okay,
iodine.
so the concentration of iodine could be changed. So if we wanted to, class, could we design several new experiments just from this one? And that's the nature of science, isn't it? We come up with a new testable question in the midst of one we're already exploring. So now let's go ahead and let's draw our stage 3 with my beaker. Adrian, what do you think?
Change the color from cornstarch being green.
Are we going to need to make the entire model black?
No.
Why not, Adrian?
Because it didn't fully change.
Okay, so we don't have full-on diffusion yet. We don't have equilibrium at this point, so we're not going to model that in our drawing. So we probably need to have a mixture of cornstarch that's still the same color as before, correct? Class, if you're with me, say I am. Okay, good. So we're going to need to have the cornstarch. Some of it didn't change, so we're going to need that there, but some of it did.
Iodine.
Okay.
Iodine.
And class, would each of your drawings be exactly the same? Is table 7's drawing going to be exactly like table 6's? Because did their bag completely diffuse one or two of those molecules? But buddy, if your bag went to town and it diffused like crazy, then you want to be honest and record those observations as such. Are we done with our model?
So just like this.
Okay. What diffused where? Okay, Everett?
The iodine used into the bag.
Okay, so how would I model that, Everett? Iodine molecule into the bag, and then I would probably want to annotate out next to my diagram what that arrow represents. Okay, so we're going to say iodine diffuse into the bag, and that's what caused the color change. So we're going to and let's answer our conclusion questions. Now that we're back in the classroom, let's make sure we understand what actually happened with our lab. Based on your observations, what substance moved? Was it iodine or was it starch? We've learned that a selectively permeable membrane is. Is this baggie selectively permeable?
Yes.
Does it let certain materials in but keep other materials out?
Yes.
Okay. So we would say, class, what would we say for that question? Is it selectively permeable?
Yes.
Yes, it is. And what substance is it permeable to? Iodine. It lets iodine in, but it keeps cornstarch inside the bag. It's not going to let it pass over that membrane. Okay. We've learned that the cell has different ways of getting materials in and out of it. And in this experiment that you guys just did, you actually saw this in action. Okay. So remember that plant or animal cells or any cell in general has a selectively permeable membrane. And what you observed was that those molecules were tiny enough to fit through that porous membrane and get in order for the cornstarch to change color. Now let's make a connection back to the cell. Why is that important for a plant or an animal cell? Why does it need this process? Why does a cell need a semi-permeable membrane? Why is that important for cell health? Okay, Jackson, say it again. The cell doesn't need too much or too little of any substance. Why not, Jackson? It could die. And if too many cells die, what will happen to the plant or animal? It dies. So class, the semi-permeable membrane is critical to cell health. It allows those materials to transfer across that cell membrane. Okay? Now, here comes the part of the lesson where you get to show me all the fabulous things that you've learned in this couple of days. I've given you on your last page of your handout another picture. So this is a totally different situation than what we've just observed and what we've seen. And it's a way for me to see if you have achieved the goal that I set for you, which was to illustrate osmosis or diffusion. Okay? So here's how you're going to do that. You're going to read the little prompt at the top of your paper, and the prompt tells us the body cell. Your task is to illustrate how those molecules would move into the cell after 3 minutes. So after 3 minutes has gone by, what would those molecules look like after they've moved? And then after 6 minutes, what would those molecules look like after they've moved? After you've done your illustrations, then you're going to come down and you're going to answer questions A and B. What is the scientific name that you could possibly add in to your writing? Jasmine?
Diffusion.
Okay, so possibly you might include the term diffusion. And Jasmine, would you want to define that term? Anytime we use a term in science, do we want to define it for our reader? Class, remember, your audience is not me. Your audience is a 3rd or 4th grade student who has no idea what we've been learning about. So you give them as much detail as possible. Be sophisticated. What is another term you can include in your writing? Equilibrium. Okay, so you might...